Hi, everybody. This is Professor Connor. I know some of you are having problems with the PowerPoint, so I'm trying to find a way to get that PowerPoint to you so that it can be used. So I'm going to record the presentation in this video. Hopefully it works, and then I will upload it and send it to everybody. So if you're getting this message, the PowerPoint is attached, and hopefully all of the sound. There is a piece that is missing. And so the contraction part, but that's just where the muscles are basically contracting back and forth for to do work so that you can move the muscle, move whatever if you're moving an object, it's the way that your muscles work with that. That piece is actually in the PDF as well. So I would peruse the contraction piece in the PDF. But otherwise, I'm going to try this. Hopefully it works. And you'll still be able to see all of the pieces, and I'm hoping the volume will come through. So I'm going to share my PowerPoint right now, hopefully. Okay, so here's the PowerPoint, and we're going to start from the beginning, and I'm just going to let it run. Hopefully it'll run all the way through, and then we'll see if it works.
This is your protocol for Lab 6, Skeletal Muscle Physiology. The things we will be learning in this week's lab are the following: review anatomy of a sarcomere, learn and understand the steps to contraction and relaxation of skeletal muscle, learn and understand the following concepts and terms: isometric and isotonic contractions, threshold stimulus, recruitment, summation, tetanus, incomplete and complete, and muscle fatigue. The items you will need for this lab are the following: the worksheet that is provided in this module, titled Skeletal Muscle Contraction and Relaxation, a Synopsis, provided by Melanie Farr. Simple light switch in a dark room connected to a lamp, a small light object, and a large heavy object. To give you a review of the anatomy of a muscle and help you picture what we are going to be talking about today in the lab, you can see that there is a picture of the femur right here, and coming off of the greater trochanter of the femur, you can see this muscle. There's a covering around the entire muscle, which is called the epimysium. That's a layer of dense, irregular connective tissue. It's pointing to this white covering on the outside. And you can notice that inside this muscle, you can see these circles. And this one that is coming out right here is representing one of those circles that you can see. And this box around here is labeled the fascicle. So a bunch of fascicles that have perimysium surrounding the outside with that dense irregular connective tissue. You bundle a bunch of those together and you have a muscle. Then if you look inside the fascicle, you're going to see a bunch more circles. Each one of these circles is a muscle fiber or a muscle cell, which you can see labeled right here. And around this entire thing here, the covering around a muscle fiber is the endomysium. And then you can see further these things coming out of the muscle fiber are myofibrils. And each myofibril has a bunch of sarcomeres inside of it. And you can see the filaments coming out of the myofibril. And then you can see down here, this yellow is indicating the sarcoplasmic reticulum. So this is a good picture to see how we are looking at the microscopic level down here of the muscles, but you can see how that fits together with the entire thing. So if you contract enough sarcomeres, that's going to contract a muscle fiber. If you contract enough muscle fibers, that's going to contract a fascicle. And then if you contract enough fascicles, that's going to contract the entire muscle. The following is a picture from your textbook. It is a picture of the sarcomere, and it includes some of the terms that you will need to remember from anatomy or learn the first time if you haven't had it. You'll see right here the actin or thin filament. You'll see right here the myosin or thick filament, the Z disc out here and out here on the periphery of the sarcomere. See the H zone right here, which would completely disappear when the muscle cell is completely contracted. You'll notice the A band here and I bands here and here. And if you remember, the A band is the dark band and the I band is the light band. And a good way to remember that is the second letter of both words dark and light are the letters that coincide with those. The first activity of this week is the muscle contraction and relaxation worksheet. Please get out your worksheet titled Skeletal Muscle Contraction and Relaxation, a synopsis, and follow along as you go through this activity. The three main steps for activity 1 for muscle contraction and relaxation are 1, excitation of the muscle cell membrane, 2, contraction, 3, relaxation. The first thing that has to happen for excitation of the muscle cell membrane is an action potential. So the action potential is going to travel down to the end of the axon, and you'll see this picture up in the corner is just a blown up version of one of these synaptic knobs. So I might be pointing to this picture up here or this one down here. So if this is a muscle cell that this is innervating, then what type of neuron does this have to be? Remember, we went over this last week in last week's lab. So that would be a somatic motor neuron. Then the next thing that happens is calcium is released from the synaptic terminals into the neuromuscular junction. So this space right here, or that's also sometimes referred to as NMJ. Then calcium binds to the receptors on the motor end plate portion of the sarcolemma. And you can see right here, the motor end plate is pointed to on this close-up here and down here. So you can see it in both views. These receptors are also acetylcholine-gated sodium channels. Opening these channels causes depolarizing graded potential and excitation of the sarcolemma. If threshold is reached, action potential begins on the sarcolemma and travels deep into the muscle fiber via the T tubules. An action potential arrives at the lateral sacs, also called terminal cisternae, of the sarcoplasmic reticulum. The last main step is relaxation. In order for the muscle fiber to relax, the action potential on the axon must end, or acetylcholine would continue to be released. Acetylcholine, already present in the neuromuscular junction, must be destroyed, or muscle receptors would continue to respond. The enzyme acetylcholinesterase destroys acetylcholine at the neuromuscular junction. No acetylcholine means no depolarizing graded potential on the muscle cell, and therefore no action potential follows on the sarcolemma. No action potential on the sarcolemma means that calcium will not be released from the sarcoplasmic reticulum. Calcium present in the sarcomere must be removed for the tropomyosin barricade proteins to go back up. This is accomplished by pumping calcium back into the sarcoplasmic reticulum and requires ATP. Now, actin-thin filaments can slip back into place and the sarcomere lengthens. Activity 2 deals with isometric and isotonic contractions. You want to understand the difference between the two. As you go through this activity, the question you want to ask yourself is, does the muscle shorten or not? If it does, that means it's an isotonic contraction. If it does not, that means it's an isometric contraction. Please take a minute to go through the steps of this activity before going to the next slide. You will need this to fill out your post-lab report. Activity 3 is demonstrating an analogy to threshold stimulus. Please take a minute to follow the directions on this slide before moving on to the next one. As you recall from your pre-lab reading, a motor unit is a single somatic motor neuron plus the muscle fibers innervated by that neuron. Take note that a somatic motor neuron can innervate a single muscle fiber, or it can innervate multiple muscle fibers. Either way, when a motor unit is stimulated, all of the muscle fibers or muscle cells within that unit will contract with all of the force they can develop. This is an all or nothing principle. So if you apply this to the activity you just did with the light switch, when you turn the light switch on, the light came on. If you turned the switch halfway up, the light would not come halfway on. It would either come on or off. It is an all or nothing. This is the same concept with motor units. Activity 3 is about recruitment. From your pre-lab reading, you will recall recruitment is the process of increasing the number of motor units that are active. Take the analogy of the light switch and now think about your whole house. Do you want to have all of the lights turn on in your whole house every time you flip a switch? Absolutely not. Your electric bill would be astronomical. So to conserve energy, You only turn on the ones you need depending on where you are in the house and how much is going on in your house. More movement throughout the house would mean the need for more lights. Also, the more visibility you need will increase the need for more light. Think of this in terms of your muscles as you complete the next activity. Please follow the steps on this slide before moving on to the next slide. You will need the answers on this activity in order to answer the questions on your post-lab. In activity 4, you will learn about summation, tetanus, and muscle fatigue. As a reminder of the definitions from your pre-lab, summation is when a stimulus is applied to the muscle fiber before it has completely relaxed from a preceding twitch. In summation, the force produced by an individual muscle fiber increases when it is stimulated to contract more frequently, which causes the twitch to add together, generating more force. Incomplete tetanus is characterized by a higher average tension than that generated by a single muscle twitch. However, the individual twitches are still observable. Complete tetanus is when the stimulus frequency is increased even further. Summation produces a plateau. No relaxation occurs. And muscle fatigue is a decline in muscle tension due to repetitive stimulation. You can think of these terms by thinking about someone feeling lazy. The lazy person is like the muscle fiber. You can convince them to get off the couch with an initial request or an action potential. If you ask that person to do another task before they sit down, they are more likely to stay and help and be more productive. This is like summation. You can continue asking for help so they accomplish more. This is like generating more force. If you ask them as they start walking back to their comfy place, you might see them start to get annoyed, but they still continue to help. This is like incomplete tetanus. Then you might switch to asking them before they finish the previous task, putting them at full capacity. This is like complete tetanus. But as we all know, this will only last so long. Once they are asked to do too much, they will go back to laying comfortably on the couch with no chance of them getting up again for quite a while. And this is like muscle fatigue. This is a visual representation to help you see the difference between a twitch in number 1, summation in number 2, incomplete tetanus in number 3, which is also referred to as unfused tetanus, and complete tetanus in number 4, which is also referred to as fused tetanus. You will notice that the red arrows indicate how frequent the stimuli is, and the waves show the amount of tension that is caused.
Okay, so just really quickly, if we go back to contraction phase, contraction of a skeletal muscle fibers initiated and maintained by the arrival of, let me just get there, pause it to make sure it doesn't move on. Return.
The last main step is,
sorry.
The last
Okay, I think that worked. So the contraction of a skeletal muscle fiber is initiated and maintained by the arrival of the action potentials at the axon terminal of a motor neuron. So upon the cessation of action potentials and the transport of calcium back into the SR, the contraction either stops and the muscles then relax. So that's part of that. So stop sharing. All right, so I know that should at least help you get through the lab. If you need more additional information, there is the PDF, and it has some of the detailed information in it that she was going over a little bit more in detail there. But again, if you still need help, let me know, and I'm here to help you. Hopefully this makes a little bit more sense, and for those of you who haven't had a chance to get it to work, this might help. So I'm sending this out. Hope you're doing well. Happy 4th of July. You do have until tomorrow night. Remember, I extended it because of the holiday to submit everything. And so, again, I might be off and on tomorrow because of the holiday and later tonight with my family. So if you need help, just email me and I'll get back to you as soon as I can. Have a great weekend, and we'll see you next week.